we interrupt you for a quick interview? It's just 23 questions.
Sure.
We've never interviewed a pair. Are you two experts up for the challenge?
Yes. So we formed great partnership here in GMP manufacturing, and we oftentimes finish each other's sentences. My name is Aga Gascoyne, and I'm the Director of GMP Upstream Manufacturing.
And I'm Ashley Craddick, Senior Director of GMP Downstream Manufacturing.
Can you share why GMP manufacturing plays such a vital role in the development of gene therapies?
Sure. At this stage in our client's journey, they rely on us for operational excellence to manufacture their drug product. That is used in their clinical trials and directly injected into their patients, so safety and quality are always at the forefront of our minds. At Forge, we have a rich GMP culture.
And we pride ourselves on adhering to current good manufacturing practices and making sure that the entire company knows what it means to have GMP culture.
Wow, you do finish each other's sentences. And you also both lead critical teams, right?
Yes, but we wouldn't be able to do that without our fabulous teams. They exemplify Forge's Hope core values, and every day when they come to work and they do manufacturing in the suite, they know what kind of impact it can have on patients' lives.
We kick off every manufacturing run by explaining to our teams who our client is, the disease that they're working on, and how the therapy impacts their patient community.
Now that is mission-driven. We like to toss in a couple of fun questions, too. Ashley, who is your dream dinner guest?
Ooh, I would have to say probably Ina Garten. Her and I would cook an amazing meal for all the women on our team.
How do we get an invite? Back to GMP, Aga, can you share how support and QC come into play to ensure a client's GMP run goes smoothly?
Yes. So our GMP support team starts with the buffer prep to make sure that all of the runs start on time. And then we also do room changeover between each run, each client run. We do cleaning, documentation, follow procedures for that. And I also do environmental monitoring done by QC to make sure that all of the rooms are ready for GMP manufacturing.
Great, that makes sense from an environment perspective. From a process perspective, how do you ensure proper documentation is ready for the client? Ashley?
We work hand in hand with our MSAT and quality teams to prepare all of our batch records that are used in the production. They get drafted, reviewed, and approved, and then all of the operators train on them. After that, we're ready to head into suite. And speaking of that, I actually have to go check on something, so I'll see you soon.
Thanks, Ashley. Aga, back to you. Can you share the benefits of engineering runs before GMP?
Yes, there are many benefits. One of them is we can apply lessons learned from engineering run to GMP run. We can also demonstrate that our runs are successful at scale, and we can make the material for initial stability studies, and there are many more other benefits.
You lead the team in upstream manufacturing. What can you tell us about the first step?
Yeah, let's go to the clean room and I can show you. Welcome to the clean room. These are GMP manufacturing suites where we perform our upstream and downstream processing. You look great in full PPE.
Thanks. I call this look sterile casual. So what's the first step in upstream manufacturing?
Because we are making our own plasmids and also we have our own master cell bank of our cell line, upstream is easy to dive into. So first, we thaw a vial of frozen cells and we scale them up in the seed train through cell culture.
I'm learning so much. What's happening behind you?
Right here?
What's in that flask?
These are our ignition cells that are about to be passaged. So we use a rolling seed train where we can scale the cells up all the way to the scale that is needed for clients' manufacturing process. These ones are going to be scaled to 1,000-liter scale.
Okay, how do you make sure the cells are healthy and ready for production?
So we monitor the cells daily, then whenever they are ready for production, we then transfect them, and then after that, we incubate them in the bioreactor until they are ready for harvest and license.
Last question while you're gowned up. Music or podcasts on road trips?
Music. Pearl Jam, Peter Gabriel, Enya, whatever the occasion.
That's quite a mix. Perfect for a bioreactor. Get it? Thanks, Aga. With even flow on our minds, steer us downstream. Let's see how Ashley and her team take it from here.
Yeah, let's go for that door to the left and you'll see it. All right, I'll see you later.
Ashley, is that you? We just learned about the upstream process. Tell me how you pick up from where Aga left off.
After lysis and harvest, the upstream team passes the material to the downstream team for depth location by clarification. After that's complete, we load it onto our single-use chromatography system using Forge's commercially viable bespoke process.
We hear a lot about empty-full separation. How does your team enrich for full capsids?
We utilize cesium chloride ultracentrifugation for our platform. This separates the empty and full capsids to the client specifications for their end material.
Fun question. You played college softball. What would be your walk-up song today?
Ooh, I would probably pick Swag Surfin' by Fly, just like my birthday twin, Aaron Judge, on the New York Yankees.
Ooh, that's a good one. Okay, we've seen most of the steps. How do we knock it out of the park?
The final step in our process is filling, which occurs in a dedicated suite. We can go check it out later. All of our filling operations are on site, which makes for a smooth transition between the downstream team and the filling operators.
How long does it take to complete filling?
This particular client's using our Watson & Marlowe semi-automated filling and needs about 100 vials, so that should take no longer than an 8-hour shift, but we can go up to 400 vials.
I think I'm getting the whole picture now.
With filling almost complete, let's hop out of our bunny suits and go meet Aga.
Filling is completed, so what's next?
Next, we take our samples and pass them off to QC for testing and inspection, and then they get stored in the warehouse long term. Hey, Aga.
Hi.
What is the most important thing to do once you finish a run?
Documentation.
The batch records are completed and reviewed immediately post-run by both the upstream and the downstream teams, and this is done in parallel with the QC testing to keep our timelines for release.
Hey Aga, how is your final documentation coming along?
Great. I'm about to pass them off to QA now.
Sounds like you've completed everything. What's next?
Room changeover.
One last fun question. For a Columbus, Ohio summer ice cream, Graders or Jenny's?
Jenny's.
And that is your 23rd question. Ashley, you mentioned that Watson and Marlowe filler. Will you show me?
Sure, but we have to get going back up.
Okay, maybe show me next time. Thanks for the tour, you two. Have a great summer.
Bye.
Bye.